Processing of tRNA. Processing of pre-tRNA involves cleavage, trimming, chemical modification, and splicing of pre-tRNA. tRNAs are commonly synthesized as precursor containing extra sequences that are removed by combination of endonucleolytic and exonucleolytic activities. The 5' end of tRNA is generated by a cleavage action catalyzed by the enzyme ribonuclease P. In E. coli, endonuclease RNase E of or E make cut at 3' end. Then RNase D, which acts as an exonuclease, trims 7 nucleotides from this new 3' end. One feature that is common to all tRNAs is the triplet sequence CCA at the 3' terminus. The triplet sequence CCA is not coded in the genome but are added as part of tRNA processing. Apart from cleavage and trimming, several types of chemical modifications like methylation, saturation of a double bond occur in the pre-tRNA. So RNase P, RNase D, and RNase E by E or F, processing of pre-tRNA. Most splicing reactions occur by transesterification reactions. The splicing of tRNA genes is achieved by a different mechanism that relies upon separate cleavage and ligation reactions. An endonuclease cleaves the tRNA precursor at both ends of intron. Release of the intron generates unusual 5' hydroxyl and 2' 3' cyclic phosphate ends. The 5' OH end is phosphorylated by a polynucleotide kinase, and the cyclic phosphate group is opened by phosphodiesterase to generate a 2' phosphate terminus and 3' OH group. Exon ends are finally joined by an RNA ligase, and the 2' phosphate is removed by a phosphatase. Introns in tRNA genes are unrelated, and there is no consensus sequence that could be recognized by the splicing enzyme. The splicing of tRNA depends principally in one recognition of a common secondary structure in tRNA. All the introns include the sequence that is complementary to the anticodon of the tRNA. The exact sequence and size of the intron is not important. The nuclease, phosphodiesterase plus kinase, then RNA ligase, then mRNA degradation. The average half-life of bacterial mRNA is only about 1.5 minutes. Degradation of mRNA occurs in the 3' 5' direction and mediated by several endo and exonucleases. No enzyme capable of RNA degradation in 5' 3' direction has yet been reported in bacteria. Endonucleases RNase E and RNase 3 make internal cut in RNA molecules, whereas exonuclease RNase 2 and polynucleotide phosphatase PNPase removes nucleotides sequentially from the 3' end of an mRNA. In E. coli, RNase E and PNPase along with RNA helicase are located within a multi-protein complex called the degradosome. The average half-life of eukaryotic mRNA is much longer than the bacterial counterpart, on average 10 to 20 minutes in lower eukaryotes like yeast to several hours in mammals. In eukaryotes, there are several degradation pathways of mRNA which occur in both 3' 5' and 5' 3' direction. In both cases, removal of poly A tail occurs first. Removal of tail is catalyzed by deadenylase enzyme. In major 5' 3' degradation pathway, deadenylation at the 3' end triggers decapping at the 5' end. This shows that each end of the mRNA influences events that occur at the other end. Decapping reaction occurs by the cleavage of 1 to 2 bases from the 5' end. Removal of the cap triggers the 5' 3' degradation pathway in which the mRNA is degraded rapidly from the 5' end by the 5' 3' exonuclease. The second degradation pathway, deadenylated mRNA are degraded by the 3' 5' exonuclease activity. Exosome, which is related to degradosome, degrades the mRNA in the 3' to 5' direction. The exosome is also found in the nucleus where it degrades unspliced precursors to mRNA. Then mRNA surveillance. In the last few years, it has become clear that a conserved mRNA degradation system referred to as mRNA surveillance exists in the eukaryotic cells to degrade apparent mRNAs. So mRNA surveillance is commonly referred to as a nonsense mediated decay or NMD because the majority of transcripts not to be degraded by this mechanism contain premature translation termination codons. This process plays an important role in checking that mRNA have been properly synthesized and functions at least in part, to increase the fidelity of gene expression by degrading apparent mRNAs that, if translated, would produce truncated proteins. A critical issue is how normal and apparent mRNAs are distinguished and how that distinction leads to differences in mRNA stability. And recent results suggest a model with three main points. First, mRNAs have a domain organization that is in part a reflection of the completion of nuclear pre-mRNA processing events. Then second, the critical aspect of distinguishing a normal form and aberrant mRNA is the environment of the translation termination codon as determined by the organization of the mRNP domains. Third, the cell distinguishes proper from improper termination through an internal clock, that is the rate of ATP hydrolysis by UPF1P. If termination is completed before ATP hydrolysis, the mRNA is protected from mRNA degradation. Conversely, if termination is slow, then ATP hydrolysis is a structural rearrangement occurs before termination is completed, which affects the fate of the terminating ribosome in a manner that fails to stabilize the mRNA. Next is about regulation of gene transcription. Prokaryotic transcriptional regulation is accomplished by a series of controlled sequence-specific DNA-protein interactions at DNA target sites located near the beginning of transcription of a gene. Two types of DNA-protein interactions are required for regulated transcription. One required DNA-protein interaction determines where transcription begins. It involves the DNA segment called the promoter and the protein RNA polymerase. An RNA polymerase binds to the promoter DNA. Transcription can initiate a few bases away from the promoter site. Hence, every gene must have a promoter or it cannot be transcribed. The other type of required DNA protein interaction regulates whether or not promoter driven transcription occurs. DNA segment near the promoter serves as a protein binding site for regulatory proteins called activator and repressor. The binding of an activator protein to its target DNA site is a necessary prerequisite for transcription to begin. Such instances are sometimes referred to as positive regulation because it is the presence of bound protein that is required for transcription. Whereas binding of a precursor protein to its target site stops the transcription to occur, referred to as negative regulation. Thus, gene regulation may be positive or negative. Negative regulation mediated by precursor repressor that block or turn off transcription, and positive regulation mediated by activator that is required for the activation of a transcription unit. The expression of some genes occur constitutively, meaning that they are expressed at a reasonably constant rate and not subject to regulation. These are referred to as housekeeping genes. Housekeeping genes need to be expressed in essentially all types of nucleated cells because they encode a key product that is required to fulfill a general function of all cells. Next is about operon model. The basic concept for how gene regulation occurs at the level of transcription in bacteria was provided by the classic model called operon model, Jacob and Monod in 1961. An operon is a unit of bacterial gene expression and regulation, including structural genes and control elements in DNA recognizing by regulator gene products. All the genes in operon are expressed as a single unit. This type of arrangement is common in prokaryotic genomes. A single promoter required to initiate transcription of the cluster is the point where expression of all of the gene is regulated. A typical example is the lactose operon or lac operon, the first operon to be discovered, which contains three genes involved in conversion of polysaccharide sugar lactose into its monosaccharide units glucose and galactose. The lac operon occupies about 6,000 base pairs of DNA. At the left, the LAC1 gene has its own promoter and terminator. The end of the LAC1 region is adjacent to the promoter P. The operator O occupies the first 26 base pairs of the transcription unit. The long LAC-Z gene is followed by the LAC-Y and LAC-A genes at a terminator. The entire system, including structural genes and the elements that control their expression, forms a common unit of regulation. This is called an operon. So the activity of the operon is controlled by regulator genes whose protein products interact with the cis-acting control elements. Here we could see a picture which depicts the general structure of an operon, lactose operon, as conceived by Jacob and Monod. The lac operon is transcribed as a multigenic or polycystonic mRNA. The Z, Y, and A indicate the LAC-Z, LAC-Y, and LAC-A loci. The mRNA transcript is translated as individual proteins. The lac operon regulator gene is decoded as I. The O stands for operator and the P for promoter. Both the operator and the regulator gene have their own promoters. So regulatory gene promoter, operator, structural genes code for the enzymes involved in lactose metabolism, alpha galactosidase, permease, transacetylase. The lac operon in E. coli is an example of inducible operon. An inducible operon is one whose expression increase in response to an inducer. An inducer is a small molecule that triggers gene transcription by binding to a regulator protein. The lac operon in E. coli is an example of inducible operon. An inducible operon is one whose expression increases in response to an inducer. An inducer is a small molecule that triggers gene expression transcription by binding to a regulator protein. In lac operon, lactose and its analogues that lead to expression of the lac genes are termed inducers, and the relief of expression by operon is termed as induction. The lac structural genes, lactose is a beta galactoside that E. coli can use for energy and as a carbon source. The function is mediated by three structural genes. The structural gene is simply any gene that codes for a protein or RNA compound. The roles of the three structural genes are LAC-Z codes for the enzyme beta galactosidase. Beta galactosidase to cleave the lactose molecule to yield glucose and galactose. This enzyme also converts lactose to allolactose. So LAC-Y codes for the beta galactoside permease which transport lactose into the cell. So LAC-A codes for beta galactoside transacetylase. The transacetylase is not essential for lactose metabolism but appears to play a role in the detoxification of compounds by transferring an acetyl group from acetyl-CoA to beta galactosides. All three genes are transcribed into a single multigenic messenger RNA molecule. Regulation of production of its mRNA coordinates the regulation of the synthesis of all three enzymes: galactose, lactose, hydrolysis of lactose, beta galactosidase, isomerization of lactose, galactose, glucose, and allolactose. The enzyme beta galactosidase hydrolytically cleaves lactose into glucose and galactose. The enzyme can also convert lactose to allolactose. Then regulatory components of the LAC system. LAC repressor. LAC repressor is a product of a regulator gene. LAC repressor is a product of a regulator gene. A regulator gene simply describes a gene that codes for a protein or RNA involved in regulating the expression of other genes. The regulatory genes are I gene and codes for the LAC repressor protein, so named because it can block the expression of the Z, Y, and A genes. The I gene happens to map fairly near the Z, Y, and A genes, but this proximity does not seem to be important to its function. 4 identical subunits with molecular weights of 38,000 assemble to a LAC repressor molecule. At any one time, only 2 subunits of the repressor appear to find to the operator, and finding occurs in the major groove double helical DNA. There are normally 20 to 40 repressor tetramer molecules in the cell. A LAC repressor is a molecule with 2 recognition sites: the DNA binding site that can recognize the specific operator DNA sequence for the lac operon, and an allosteric site that binds the lactose allosteric effector and similar molecules. Analogues of lactose. The DNA binding site of a DNA LAC repressor is able to bind with high affinity to only one DNA sequence in the entire E. coli genome, the LAC operator. The specificity of high affinity DNA binding ensures that the repressor will bind only to the site on the DNA near the genes that is controlling and not to random sites distributed throughout the chromosome. By binding to the operator, the repressor prevents transcription by RNA polymerase that has bound to its LAC promoter site. So, as already mentioned, the allosteric site of the LAC repressor binds to lactose and structurally similar molecules, lactose analogues, and the repressor binds to lactose or its analogues. The protein undergoes a conformational allosteric change. The site alteration in space changes the DNA binding site, so the repressor no longer has high affinity for the operator. Thus, in response to binding lactose, the repressor falls off the DNA. Then operator is the cis-active site on the DNA to which the LAC repressor binds. It is located between the promoter and the Z gene near the point at which transcription of the mutagenic mRNA begins. The transcription between the LAC operator site on the DNA and LAC repressor is crucial to proper regulation of the LAC operon. The negative regulation of LAC operon. The LAC genes are controlled by negative regulation in the absence of an inducer, lactose or an analog. The LAC repressor binds to the LAC repressor operator site and prevents transcription of the LAC operon. Most of the effect of LAC repressors binding to the operator is to block the progression of RNA polymerase transcription. In this sense, the LAC repressor molecule is a negative regulator and acts as a roadblock on the DNA. Consequently, all of the structural genes of the LAC operon are repressed and there is no beta-galactosidase, beta-galactoside permease or transacetylase in the cell. How does the presence of inducer lactose trigger the expression of LAC operon? When an inducer is present, it binds to the allosteric site of the LAC repressor, thereby inactivating the operator DNA binding site of the LAC repressor protein. However, lactose itself does not have this effect, rather allolactose does. Allolactose is that referred to as the inducer of the LAC operon. Formation of the allolactose is also catalyzed by the beta-galactoside, a lactose analog that is capable of inducing the LAC operon while not itself serving as a substrate for beta-galactosidase is an example of a gratuitous inducer. An example of this one is the isopropythiogalactosidase IPTG. Inactivation of operator by the LAC repressor permits the introduction of transcription of the structural genes of the LAC operon, and through the translation of the multigenic mRNA, the enzymes beta-galactosidase, beta-galactoside permease, and transacetylase now appear in the DNA cell in a coordinated fashion. Then LAC repressor. LAC repressor is a product of a regulator gene. A regulator gene simply describes a gene that codes for a protein or an RNA involved in regulating the expression of other genes. The regulatory gene I gene encodes the LAC repressor protein, so named because it can block the expression of X, Y, and A genes. And the I gene happens to map fairly near the X, Y, and A genes, but this proximity does not seem to be important to its function. 4 identical subunits with molecular weights of 38,000 assembled to a LAC repressor molecule. At any one time, only 2 subunits of the repressor appear to bind to the operator, and binding occurs in the major groove double helical DNA. There are normally 20 to 40 repressor tetramer molecules in the cell. The LAC repressor is a molecule with 2 recognizable sites, a DNA binding site that can recognize the specific operator DNA sequence for the LAC operon, and the allosteric site that binds the lactose allosteric effector and similar molecules, analogs of lactose. The DNA binding site of the LAC repressor is able to bind with high affinity to only one DNA sequence in the entire E. coli genome, the LAC operator. The specificity of high affinity DNA binding ensures the repressor will bind only to the site on the DNA near the genes that is controlling and do not random sites distributed throughout the chromosome. By binding to the operator, the repressor prevents transcription by RNA polymerase that has bound to its LAC promoter site. As already mentioned, the allosteric site of the LAC repressor binds to lactose and structurally similar molecules, lactose analogs. Then the repressor binds to lactose or its analogs. The protein undergoes a conformation and allosteric change. This slight alteration in space changes the DNA binding site so that the repressor no longer has high affinity for the operator. Thus, in response to binding lactose, the repressor falls off the DNA. Then operator. The operator is the cis-acting site on the DNA to which the LAC repressor binds. It is located between the promoter and the Z gene near the point at which transcription of the multigenic mRNA begins. The interaction between the LAC operator site on the DNA and LAC repressor is crucial to proper regulation of the LAC operon. A negative regulation of LAC operon. The LAC genes are controlled by negative regulation. In the absence of an inducer, lactose, or an analog, the LAC repressor binds to LAC operator site and prevents transcription of the LAC operon. Most of the effect of LAC repressors binding to the operator is to block the progression of RNA polymerase transcription. In this sense, the LAC repressor molecule is a negative regulator and acts as a roadblock on an DNA. Consequently, all of the structural genes of the LAC operon, the X, Y, Z, Y, and A genes are repressed, and there is no beta-galactosidase, beta-galactoside permease, or transacetylase in the cell. How does the presence of inducer lactose trigger the expression of lac operon? When an inducer is present, it binds to the allosteric site of the lac repressor, thereby inactivating the operator DNA binding site of the lac repressor protein. However, lactose itself does not have this effect, rather allolactose does. Allolactose is thus referred to as the inducer of the lac operon. Formation of allolactose is also catalyzed by the beta-galactosidase, the lactose analog that is capable of inducing the lac operon while not itself serving as a substance for beta-galactosidase is an example of gratuitous inducer. An example is isopropyl thiogalactoside. In the absence of lactose, transcription blocked. But in the presence of lactose, introduction of lac operon. In the absence of lactose, the lac repressor binds DNA and represses transcription from the lac operon. Lactose binds to the lac repressor, leading to its dissociation from DNA and to the production of lac mRNA. Then constitutive expression of lac operon. Constitutive mutants are mutants in which the three lac operon genes are transcribed at all times, that is, they are not turned off even in the absence of lactose. And the LAC-I gene has been mutated so that its product LAC-I is not capable of binding to operator DNA. The organism will exhibit constitutive expression of the lac operon. Similarly, mutations in lac operon locus such that the operator sequence will not bind a normal repressor molecule constitutively express the lac operon genes. Both types of mutants produce the same phenotype. So, constitutive expression of the three lac operon genes. The regulator constitutive mutants are designated. The operator constitutive mutants are designated. Expression of lac operon in merozygotes. Partial diploids in E. coli can be created through sexduction because some strains of E. coli have the lac operon incorporated into an F factor. Since F negative strains can pass the F particle into F negative strains, lac operon diploids, also called merozygotes or partial diploids, can be formed. So, case 1. A lac operon in E. coli with a mutation of the regulator site is shown. The cell has a constitutive phenotype. The operator cannot bind the mutant type repressor protein and thus transcription is continuous even in the absence of lactose. Then E. coli chromosome is there. Mutant repressor not able to bind operator, constitutive. Then the wild type regulator gene is introduced in an F factor where there is both a bacterial chromosome and an F factor, each containing a regulator gene. In this case, the phenotype is now normal or inducible because enough repressor is produced by the F allele to bind to both operators. So, E. coli chromosome, normal repressor able to bind operator, both operons repressed. A lac operon in E. coli with a mutation of the operator. The cell has a constructive phenotype. The operator cannot bind the wild type repressor protein and thus transcription is continuous even in the absence of lactose. So, there is both a bacterial chromosome and an F factor, each containing an operator. The F operator does not change the phenotype of the cell because the wild type operator exerts no control over the chromosome operator and exerts a cis dominant effect. The E. coli chromosome constitutes a normal repressor able to bind operator and repressed state. Positive regulation of lac operon. Bacteria have distinct preferences among potential carbon sources that they are offered if they are offered a choice. When glucose is available as an energy source, it is used in preference to other sugars. So when E. coli finds, for example, both glucose and lactose in the medium, it metabolizes the glucose and represses the use of lactose. The ability of glucose to inhibit the synthesis of certain enzymes, referred to as glucose effect, was recognized in bacteria at an early date by Monod. He observed that when E. coli encounters both glucose and lactose, for example, it metabolizes the glucose first and represses the use of lactose, resulting in dioxic growth, DIAUXIC. This phenomenon is due to the repressive effect of glucose on the synthesis of enzymes required for the metabolism of other sugars. The term catabolite repression was introduced as a general name of the glucose effect. Studies on catabolite repression brought about the recovery of positive control system of transcription by cyclic AMP and the cyclic AMP receptor protein CRP, also called the catabolite gene activator protein, and led to the concept that catabolite repression is caused by reduction of the level of intracellular cAMP. CRP is a positive regulator protein activated by cyclic AMP. It is needed for RNA polymerase to initiate transcription of many operons of E. coli. Cyclic AMP is synthesized by the enzyme adenylate cyclase, and its concentration is related to glucose concentration. So when the glucose level in the cell is high, the cAMP concentration is low. So conversely, when the glucose level is low, the cAMP concentration is high. E. coli and many other bacterial species contain a protein called the cAMP receptor protein that it is encoded by a gene called CRP. Mutants of either CRP or adenylate cyclase genes are unable to synthesize LAC mRNA, indicating that both CRP function and cAMP are required for LAC mRNA synthesis. CRP and cAMP bind together to form a complex denoted cAMP-CRP that is a regulatory element for the LAC system. The cAMP-CRP complex must be bound to a specific base sequence called an activation site in the promoter region in order for transcription of the LAC operon to occur. The CRP binding site is adjacent in the promoter and centered on C-61. CRP introduces a greater than 90-degree bend to DNA at its binding site. Large bend on DNA allows CRP to contact the RNA polymerase at the promoter and start transcription from start site. And molecular depiction: the dioxic growth on a mixture of glucose and galactose. Glucose represents synthesis of beta-galactosidase. After glucose is exhausted, a lag occurs until beta-galactosidase is synthesized, and then growth can resume on lactose, but at a slower rate. So relative tendency: growth on glucose exhausted, growth on lactose. Thus, the CRP complex is a positive regulator in contrast to the repressor, a negative regulator. The LAC operon is therefore regulated both positively and negatively. Effect of the concentration of glucose, lactose, and cAMP in the growth medium on the level of transcription from the E. coli LAC operon. The arrow indicates the direction of transcription. Then tryptophan operon system. The tryptophan operon required for the synthesis of an amino acid tryptophan. It is an example of a negatively controlled repressible operon. A negatively controlled operon is one that is inhibited by a regulatory protein such as repressor. Tryptophan operon is also an example of repressible operon because it is inhibited when an effector molecule is present. The tryptophan operon consists of 5 structural genes: TRPE, TRPD, TRPC, and B and A that code for enzymes involved in the conversion of choreosmic acid to tryptophan. The controlling site in TRP operon lie next to TRPE and consist of a promoter site TRPP, an overlapping operator site TRPO, and a leader region TRPL that codes for a leader peptide and for an RNA attenuator. The leader peptide is involved in the attenuation mechanism. In this repressible operon, the product of TRP gene, the repressor, is inactive by itself, called the aporepressor. It does not recognize the operator sequence of the TRP region. The repressor only becomes active when it combined with the tryptophan. Hence, in the absence of tryptophan, the aporepressor inactive regulatory protein has no effect on the tryptophan operon. Consequently, the enzyme for synthesizing the amino acids are produced and the cell makes tryptophan. Tryptophan is thus referred to as a co-repressor since it acts as an effector to convert the aporepressor into the active repressor when tryptophan binds to the aporepressor. It induces a conformational change that converts the regulatory protein into the tryptophan holorepressor. The holorepressor binds the TRP and blocks the attachment of RNA polymerase to TRP. This shuts off the transcription of the structural gene. The repressor-co-repressor complex binds up the operator and prevents the transcription of the TRP operon in E. coli. Without the co-repressor, the repressor cannot bind and therefore transcription is not prevented. Then next topic is transcriptional attenuation. An additional mechanism, transcriptional attenuation, regulates the expression of TRP operon by controlling the ability of RNA polymerase to continue elongation past specific sites. The TRP operon is regulated in part by a repressor that, when bound to tryptophan, blocks transcription. However, transcription attenuation provides an additional level of control that results in both more stringent regulation that could be achieved by reduction of initiation alone. The site of attenuation is located 162 nucleotides downstream of the transcription start site. If tryptophan is abundant, most transcription terminates at this site. Only if tryptophan is scarce does transcription continue to yield functional TRP mRNA. Attenuation of the TRP operon. The leader sequence is a stretch of nucleotides between the operator and the structural genes, and the supply of tryptophan is low. The entire operon, including the leader sequence, is transcribed into mRNA, and the supply of tryptophan is high. Only 140 bases, the part of the leader sequence that precedes the attenuator, is transcribed into mRNA. Structural genes are not transcribed. And the mechanism of attenuation depends on the fact that transcription bacteria is coupled with the translation and bacteria is coupled with the transcription. So ribosome begin translating the 5' end of an mRNA while it is still being synthesized. Thus, the rate of translation can affect the structure of the growing RNA chain, which in turn determines whether further transcription can continue. Transcription termination is signaled by a stem loop structure that forms by complementary base pairing between two specific sequences of the growing TRP mRNA chain. This structure forms if translation of the growing chain is proceeding at a normal rate, as it does when tryptophan is present in adequate supply. If tryptophan is scarce, however, protein synthesis stalls at a critical region of the message. If this occurs, the ribosome bound to the mRNA block formation of the transcription terminating stem loop, allowing TRP mRNA synthesis to continue. The critical region of TRP mRNA contains two adjacent tryptophan codons, so the rate of translation is highly dependent on tryptophan levels. This is the link between transcriptional attenuation and the availability of tryptophan. If tryptophan levels in the cell are low, the ribosome stalls in this point and transcription of TRP mRNA continues. If tryptophan is abundant, translation continues, the transcription is terminated. Then high tryptophan level, growing polypeptide chain, etc. can be seen. Then stem loop between regions 3 and 4 signals termination of transcription. Low tryptophan level, growing polypeptide chain, ribosome stalls at region 1 and alternate stem loop between regions 2 and 3. So this shows mechanism of attenuation in the E. coli TRP operon. The ribosome attempting to translate the leader transcript under conditions of excess tryptophan. The 3 to 4 stem loop forms the terminator stem, terminating transcription. Under conditions of tryptophan starvation, the ribosome is stalled and the stem loop 2-3 forms, allowing continued transcription. To predict the effects of deleting the following agents in the RNA. The gene encoding lac repressor. Cells will express beta galactosidase, lac permease like that. Then next is about bacteriophage lambda. A lambda genome is a linear double-stranded DNA of 48.5 kilobase and some 50 genes. The genome of phage lambda are contained within the protein coat of the phage. The phage lambda particle infects E. coli cells by attaching to a protein on the cell surface via its tail and projecting its chromosome to the bacterium. Specifically, lambda phage interact with the E. coli MAIB protein, which ordinarily transports maltose into the bacterium when it is available in the growth medium. After infection by the phage lambda chromosome, the bacterial cell can be made to follow one or two alternative pathways. There are also called lytic and lysogenic pathways. So in the lysogenic state, the prophage genome is inactive or repressed. In the lytic phase, the phage genes for reproduction of the active induced lambda phage have an operon type system controlling its two functional states. When normal bacteria are infected by wild type lambda phage particles, two possible sequences may follow. The phage may be integrated into the bacterial chromosome as an inert prophage, thus lysogenizing the bacterial cell, or the phage may produce a necessary enzyme to guide the production of the products needed for phage maturation and cell lysis. The phage may produce the necessary enzyme to guide the production of the products needed for phage maturation and cell lysis. The switch between these two states is mediated by proteins encoded by the bacteriophage genome. At the heart of the system are two gene regulatory proteins synthesized by virus, the lambda repressor protein and the CRO repressor. These proteins represent each other's synthesis. In the lysogenic life cycle, gene CI from the lambda phage chromosome encodes lambda repressor. The repressor acts as both a positive and negative regulator of gene expression. Lambda repressor protein is 236 amino acids in size and folds into a structure resembling dumbbell with two domains separated by a string of 40 amino acids. The repressor exists primarily as a homodimer. The carboxyl terminal domains interact strongly. The Lytic cycle second phase regulatory protein CRO is synthesized from CRO gene. CRO protein also binds specific DNA regions, the same ones as does the lambda repressor, but co-binding has the opposite physiological effect. In lysogenic state, the lambda repressor occupies the operator, blocking the synthesis of CRO and also activating its own synthesis. In the lytic stage, the CRO protein occupies a different state in the operator, blocking the synthesis of repressor but allowing its own synthesis. In lysogenic state, role of CI repressor, lambda repressor then stimulates CI repression like that. A lytic stage, role of CRO repressor, lambda CRO. So, a simplified version of transcriptional switching between bacteriophage and lambda. Within the lambda phage chromosome, the genes CI and CRO are adjacent. The two genes are transcribed in opposite directions and do not overlap. Between two genes are ATBP regions containing promoters and operates for each gene, including the binding sites for both the lambda repressor and the CRO protein. The two genes have their own promoters, that the CI promoter is known as PRM and the CRO promoter is known as PR. PR governs the rightward transcript. The two promoters are adjacent but not overlapping. If RNA polymerase binds the PR promoter, CRO mRNA will be produced. If the polymerase binds to PRM, repressor will be produced. However, the two promoters can never be occupied simultaneously. Lambda repressor and CRO are never transcribed at the same time. So, transcription of the lambda control agents in lytic and lysogenic growth. Arrows indicate which promotes the actives at the intercept period during lytic and lysogenic growth respectively. The arrows also show the direction of transcription from each promoter. And the decision between lysis and lysogeny is therefore determined by the outcome of race between C1 and CRO. If C1 repressor is synthesized more quickly than the CRO repressor, then genome expression is blocked and lysogeny follows. However, if CRO wins the race, then the CRO repressor blocks the CA repressor before enough CRO repressor has been synthesized to silence the genome. As a result, the phage enters the lytic infection cycle. The decision appears to be random, depending on chance events that lead to either the CI or the CRO repressor accumulating the quickest in the cell. In the lysogenic state, the lambda repressor controls its own synthesis from the promoter PRM by interacting with three operators. These operators have different affinities for the lambda repressor. When there is no little lambda repressor, only the OR1 site, which blocks CRO expression, is filled with the CRO protein, prevents synthesis of lambda repressor by binding to OR3. The binding of repressor to OR1 facilitates the binding of second molecule of a repressor to OR2. When lambda repressor binds to OR2, it serves to activate transcription of the CI1 gene. However, when too much lambda repressor is present, then binding to the low affinity OR3 blocks further transcription of the C1 gene.